Dear friends, in last lecture we have discussed about the human excretory system and the structural and functional unit of human excretory system or kidney, that is the structure of nephron. This nephron is made up by the two components, that is glomerulus and renal tubule. The anterior portion of renal tubule, it is the Bowman's capsule. When it associated with the glomerulus form the Malpighian body, then it leads into the proximal convoluted tubule, then loop of Henle, distal convoluted tubules, and distal convoluted tubules with the help of terminal duct opens into the common collecting duct. This common collecting duct joins and forms the large duct, it is duct of Bellini, and duct of Bellini opens into the in papillae. Papillae opens into minor calyces into major calyces, and major calyces opens into pelvis. You know that. Now after completion of this structure, now we want to focus on the most important event, that is the urine formation, or it is termed as a physiology of excretion, mechanism of urine formation. This is the most important and vital part of this topic, that is excretion and osmoregulation, how urine will be formed within the kidney. So we want to focus on this physiology of excretion or mechanism of urine formation. Generally, this physiology of excretion or mechanism of urine formation takes place with the help of three steps. That is, the first it is termed as ultrafiltration, second term that is selective reabsorption, and third process is tubular secretion, also termed as augmentation. So these are the three steps, that is ultrafiltration, selective reabsorption, and tubular secretion. Now where it occurs, generally ultrafiltration occurs in the Malpighian body of the nephron, selective reabsorption occurs in the renal tubule, all parts of the renal tubule, while tubular secretion occurs mainly in DCT, but some part also occurs in the PCT. Generally these three steps explained by the three different scientists. The ultrafiltration mainly explained by the scientist, that is Ludwig, then selective reabsorption by the Richardson, while the tubular secretion by the Hedenchen. हा चार्ट एकदा विचारलेला आहे, स्टेप्स दिलेले आहे कॉलम फर्स्ट मध्ये, कॉलम सेकंड मध्ये साइट्स आणि सायंटिस्ट तुम्हाला आयडेंटिफाय करायचे आहेत, त्यामुळे शॉर्ट मध्ये हा नीट पॉइंट साठी लक्षात ठेवायचा आहे. नाउ वी वांट टू गो इन डिटेल्स ऑफ द ultrafiltration, selective reabsorption, and tubular secretion. Before going into detail, remember one point, that is blood supply to kidney. Blood supply to kidney, generally it is average 600 ml blood supply to kidney per minute. So average it is 1200. So to complete the entire circulation of the one time blood, it is 4 minutes are required, because average volume of blood in the body it is about 5 liter. So remember this point, 600 ml blood supply to each kidney per minute, means both kidney receives 1200 ml of blood supply. It is about 20 to 25% of cardiac output. It is 20 to 25% of cardiac output. What is cardiac output? Cardiac output is the amount of blood pumped by the heart per minute, is termed as a cardiac output. And in normal person, you know that cardiac output is about 5 liter, means 5000 ml. So generally to complete the purification of entire blood for one time needs 4 minute times. So 4 minutes required for complete circulation of blood through kidneys. So हा सुद्धा ऑब्जेक्टिव्हला लक्षात घ्यावा लागेल पॉइंट्स. नाव फर्स्ट कॉन्सन्ट्रेट ऑन दिस फर्स्ट स्टेप, that is ultrafiltration. This ultrafiltration, you know that site of ultrafiltration is a Malpighian body, and that's why exact this ultrafiltration occurs in between the visceral layer of Bowman's capsule, in the visceral layer of Bowman's capsule, and you know that this visceral layer of Bowman's capsule lined by special cells, it is termed as a podocytes. So this is the podocytes. So these are the podocytes. What is podocytes? Podocytes are the modified squamous epithelial cells. So these are the podocytes which lines the visceral layer. And second, that is the glomerular capillary. The capillary present in the glomerulus. So it is the glomerular capillaries, and you know that glomerular capillary lined by endothelium in squamous epithelium, but squamous epithelium of glomerular capillary is termed as endothelium. So and this glomerular capillary consists the blood, and this blood get filtered out through microscopic pores, means through this membrane, means it is the membrane through which ultrafiltration occurs, and as a result we can observe the content here. It is termed as glomerular filtrate, or it is termed as deproteinized plasma, or it is termed as primary urine. So as a result of ultrafiltration, we can observe the glomerular filtrate, deproteinized plasma, or primary urine, means blood get filtered out through this membrane, means it is filtered out between the glomerular capillary and the visceral layer of Bowman's capsule and enters into the urinary space. Now we want to go in details of ultrafiltration. Now what is ultrafiltration? So ultrafiltration is the process of filtration of blood under high pressure at microscopic pores of glomerular and Bowman's capsule. So in simple word, ultrafiltration is the process of filtration under high pressure at microscopic pores of glomerulus, So this is the definition of ultrafiltration. So in simple word, ultrafiltration is the filtration of blood under high pressure at microscopic level of the glomerular capillaries and the visceral layer of Bowman's capsule. Now concentrate here. It occurs due to diameter difference of afferent arteriole and efferent arteriole. Mainly it is occurs due to the diameter difference of afferent and efferent arteriole. So remember, this is most important point, means under high pressure. So how it is pressure created? So for that purpose, remember few terminology. First, that is the GHP. That is GHP. What is GHP? It is glomerular hydrostatic pressure. So what is GHP? Glomerular hydrostatic pressure. This glomerular hydrostatic pressure created due to the diameter difference of afferent and efferent arteriole, and in kidneys this glomerular hydrostatic pressure is about 55 mmHg. So this glomerular hydrostatic pressure in kidney it is 55 mmHg. This is also an objective, glomerular hydrostatic pressure. Now this pressure get opposed by opposing pressure, means opposing pressure. So opposing pressure created by two type of pressures, that is first BCOP. BCOP stand for blood colloidal osmotic pressure, blood colloidal osmotic pressure, that is BCOP. Means you know that osmotic pressure in blood, it is maintained by the serum albumin. So albumin make the oppose here, and as a result blood colloidal osmotic pressure, it is about 30 mmHg. It is about 30 mmHg. So BCOP is becomes 30 mmHg. 30 mmHg. And second opposing pressure, that is capsular hydrostatic pressure. Capsular hydrostatic pressure, it is termed as a CHP, and CHP, it is capsular hydrostatic pressure, it is about 15 mmHg, means pressure exerted by the already filtrated content which is present in urinary space, it also opposes the filtration pressure. So it is termed as a CHP. So it is CHP is equal to 15 mmHg. 15 mmHg. So opposing pressure is made up by blood colloidal osmotic pressure and capsular hydrostatic pressure, while the exact pressure exerted, it is glomerular hydrostatic pressure. And from this we want to focus on the EFP, means it is effective filtration pressure. Effective filtration pressure, and this EFP is calculated by GHP minus OP. So GHP minus BCOP plus CHP. So here it is 55 mmHg, 30 plus 15. So here it is 55 minus 45. So it is 10 mmHg. Most important part. So that is effective filtration pressure in kidney, it is 10 mmHg. It is 10 mmHg. So this is the effective filtration pressure, and this effective filtration pressure is responsible to filter out filtrate. Under this effective filtration pressure, filtrate occurs. It is termed as effective filtration rate. Effective filtration rate, it is EFR, and this EFR in normal person, it is 125 ml per minute. It is 125 ml per minute. हा सुद्धा एक ऑब्जेक्टिव्ह तयार होतोय. व्हॉट इज द इफेक्टिव्ह फिल्ट्रेशन रेट? इट इज 125 ml per minute, means about 180 liter per day. Means within 24 hours we can observe 180 liter primary urine or the deproteinized plasma will be filtered out into the renal tubules of the both kidneys. So here it is effective filtration rate, it is 125 ml per minute. हा पॉइंट लक्षात ठेवावा लागेल, म्हणजे तुम्हाला इथे रेट कॅल्क्युलेट करावा लागेल. ॲज अ रिझल्ट तुम्हाला हे ऑलरेडी इथे माहीत असायला पाहिजे की विच आर द डिफरंट कंपोनंट्स ऑफ ब्लड. You know that blood consists composition of blood, that is 90 to 92% water, 90 to 92% water, then 7 to 8% organic substances, then 1 to 2% inorganic substances, then here it is some dissolved respiratory respiratory gases, and few toxic substances are present in the blood. In organic substances, you know that there is presence of nutrients in the simple form like the monosaccharides, proteins present in the simple form, that is amino acid, fat present in the fatty acid, and glycerol. There is presence of vitamins, water soluble vitamins, fat soluble vitamins. There is presence of organic substances which works as a nitrogenous waste, means there is presence of ammonia, urea, uric acid. Some creatinine is also present. In organic substances, there is monovalent bivalent ions are present, means Na plus, K plus, Cl minus, bicarbonate ion, HCO3 minus, sodium chloride. Like this, inorganic substances also present. Dissolved respiratory gases means oxygen, carbon dioxide, and some toxin substances like penicillin also present in blood, because in the process of urine formation we want to purify the blood. So all these components of blood get filtered out except it is most important, except that is plasma proteins and blood cells. Plasma proteins and blood cells means the components of blood get filtered out except हे दोन सोडून कोण, प्लाज्मा प्रोटीन्स अँड ब्लड सेल्स. यू नो दॅट देर आर 3 टाइप्स ऑफ ब्लड सेल्स, RBCs, erythrocytes, WBCs, leukocytes, and third blood platelets or thrombocytes. While plasma protein, basic 3 plasma proteins, serum albumin, serum globulin, and fibrinogen. So these plasma proteins and blood cell does not filter during the ultrafiltration, while remaining all components of blood get filtered out during ultrafiltration, because these ultrafiltration process is a non-selective. It is non-selective and passive process. Non-selective and passive process. It is most important. Non-selective means here we cannot concentrate on the essential and non-essential components of the blood which get filtered out. So it is termed as a non-essential, means essential as well as non-essential component get filtered out. Is passive process. Passive process means here movement occurs from the higher concentration to lower concentration. So the criteria used during the ultrafiltration, only the size of molecules or particles. Size of molecules or particles is the basic criteria used for ultrafiltration, means the components of the blood which have the small molecular size than the slit size or pore size. पोअर साइज स्लिट साइजच्या पेक्षा कमी ज्यांचा डायमीटर आहे, कमी ज्यांची साइज आहे तेच मोलेक्यूल फिल्टर होतील. ज्यांची साइज त्या पोअरच्या साइजपेक्षा मोठी आहे ते फिल्टर्ड आउट होणार नाहीत. म्हणजेच प्लाझ्मा प्रोटीन आणि ब्लड सेलची साइज ही या पोअर्सच्या साइजपेक्षा मोठी आहे, म्हणून प्लाझ्मा प्रोटीन्स आणि ब्लड सेल फिल्टर्ड आउट होत नाहीत. बट रिमेंबर हियर वन पॉइंट, इफ the bacterial or viral infection occurs in the glomeruli, condition is termed as a glomerulonephritis or Bright disease, in which the size of glomerular slit increases and sometimes blood cells enters into the urine, while sometimes plasma proteins enters into the urine. The condition is termed as a hematuria or proteinuria. Hematuria or proteinuria, but these are the abnormal condition. In normal condition we cannot found the plasma proteins and blood cells in urine because they does not get filtered out during the process of ultrafiltration. So as a result of ultrafiltration we can observe the glomerular filtrate. It is also termed as a deproteinized plasma. D means remove proteins from plasma, means there is presence of plasma without protein, means it does not contain blood cells. It is also termed as a primary urine, and the rate of production, it is termed as a effective filtration rate, EFR. It is 125 ml per minute, means about 180 liter per day. Imagine the content. इथे सगळ्यात महत्त्वाचं आहे, म्हणजे जर 180 लिटर युरिन पहिल्या स्टेप्स मध्ये तयार होत असेल तर तुमच्या लक्षात येईल की आपण एवढं युरिन इलिमिनेट करत नाही. आपण युरिमिनेट करतोय बिकॉज युरिन प्रोडक्शन पर डे इज अबाउट 1.5 to 1.8 liter. 1.5 to 1.8 liter, means when we compare with this 180 liter, this content is only about 1%, means out of glomerular filtrate 1% content get released out as a urine, means remaining 99% content reabsorbed into blood from filtrate, and so that the second process occurs, it is termed as a selective reabsorption. After completion of first process of urine formation, that is ultrafiltration, second process we want to focus, that is selective reabsorption. You know that as a result of ultrafiltration we can observe the glomerular filtrate. It is also termed as a deproteinized plasma. It is also termed as a primary urine. Average amount of primary urine produced per minute, it is 125 ml per minute, while 180 liter per day. Average urine output from a normal person, it is 1.5 to 1.8 liter, means only 1% content of glomerular filtrate is removed as a urine, but remaining 99% reabsorbed back by the process termed as a selective reabsorption. Selective reabsorption is the process of reabsorption of essential selective substances from the renal filtrate into the blood of peritubular capillary, termed as a selective reabsorption. Generally, so this selective reabsorption is a process occurs between the filtrate which is present in the renal tubule. From this filtrate, reabsorption occurs into the blood which is present in the capillaries surrounds the renal tubule. It is termed as a peritubular capillary. So सगळ्यात महत्त्वाचा पॉईंट इथे आहे की कुठून कुठे रिएब्सॉर्प्शन होणार आहे. री म्हणजे परत, मगाशी ब्लडमधून रिनल फिल्ट्रेटमध्ये आलेला कंटेंट पुन्हा ब्लडमध्ये येणार आहे म्हणून शब्द वापरला आहे री मीन्स अगेन absorption. So reabsorption, selective reabsorption. Why word use selective? Because non-essential component does not reabsorb back. It is eliminated as a waste. Only selected essential substances get reabsorbed. So concentrate on the substances present into the blood. So components of blood which get reabsorbed are broadly divided into three groups. That is high threshold substances, second that is low threshold substances, and third non threshold substances. So concentrate on these three different type of substances, that is high threshold substances. High threshold substances means substances of blood which are present into the renal filtrate, requires to the body 100%, means elimination of these substances occurs, it responsible for disorder. So they are termed as a high threshold substances like glucose, amino acids, vitamins. These are the high threshold substances. Then low threshold substances, the substances of renal filtrate, needs or reabsorbed on the basis of their requirement, excess eliminated from the body, while the essential percentage of substances reabsorbed back. So it is termed as a low threshold substances like water, then Cl minus ion, K plus ions, Na plus ions. So this is the low threshold substances, while the non threshold substances means these substances does not need to the body, it is eliminated as it is. So low non threshold substances like creatinine, hipparic acid, toxins, penicillins, like these substances are termed as a non threshold substances, means these substances does not reabsorb back So all components which are filtered during the ultrafiltration categorized into three types, that is high threshold substances, low threshold substances and non threshold substances. Generally the ways of selective reabsorption, generally this selective reabsorption occurs by three ways, that is first passive absorption, and second it is termed as a active absorption. So passive absorption and active absorption, these are the two basic ways of selective reabsorption. Means passive absorption, where the reabsorption of substances occurs from the higher concentration to lower concentration, while active absorption, the absorption occurs from the lower concentration to higher concentration, and here use of energy, means here it is from transport from higher concentration to lower concentration, while here it is from lower concentration to higher concentration. Here does not need energy, while here need of energy, while one process occurs here, it is osmosis. Water reabsorbed osmosis, means they can transfer from the lower concentration to higher concentration to lower concentration to higher concentration through semi-permeable membrane, that is the osmosis. So this is the ways of selective reabsorption. Now site of selective reabsorption sites, that is first, that is PCT, second loop of Henle, third that is DCT, and fourth that is collecting tubule. So these are the four sites, out of which this is PCT is the main site of reabsorption. Other are the means associated with the little amount of reabsorption, because in this PCT, if we observe 100% glucose, 100% amino acid, 100% of vitamin C reabsorbed from the PCT, then 75% water reabsorbed from DCT, then it is 70% salts or ions are reabsorbed from the PCT. So PCT is main site of reabsorption. This arrow indicates, means this broad arrow indicates the active reabsorption, while this is the passive reabsorption. So active reabsorption and passive reabsorption, through which they get filtered. So the content, when we focus on here, it is the associated part. So selective reabsorption occurs mainly in PCT. From PCT it goes to the loop of Henle, because the content, now remember here three terms, that is the isotonic, hypertonic and hypotonic. Now the filtrate, when we compare to this, means filtrate compare with plasma of blood, if they have same osmotic concentration, then it is termed as a isotonic. If this filtrate is, means plasma of blood and filtrate, means here it is filtrate, have more osmotic concentration than blood, then it is termed as a hypertonic, while less osmotic concentration than blood, then it is termed as a hypotonic. So these are the three conditions: isotonic, hypotonic and hypertonic and hypotonic. So in first part, this selective reabsorption, basically there are three type of substances get filtered out: high threshold substances, low threshold substances, non-threshold substances. Ways of selective reabsorption: passive reabsorption and active reabsorption. And site PCT, loop of Henle, DCT, collecting duct. Main site, that is the PCT, while the solution or filtrate, it is isotonic, hypertonic and hypotonic. In next lecture we want to proceed for further how the selective reabsorption occurs in loop of Henle, DCT and collecting tubule. As a result of ultrafiltration, we can observe the glomerular filtrate. This glomerular filtrate enters into the PCT. In PCT, this glomerular filtrate shows isotonic nature with the plasma of blood, So the filtrate present in proximal convoluted tubule is isotonic to the plasma. Maximum amount of selective reabsorption occurs in the PCT, and now this content move towards the descending limb. In descending limb means near about 75% content reabsorbed in PCT. Now this renal filtrate enters into the descending limb which is thin walled, permeable to water, impermeable to salt. So water reabsorption occurs from descending limb. About 10% water reabsorption occurs from descending limb, and content becomes hypertonic. This hypertonic filtrate now enters into the ascending limb. In ascending limb, it is thick walled, permeable to salt, impermeable to water. So as a result, now content again becomes hypotonic. Content becomes hypotonic to the plasma. This hypotonic content enters into the DCT. You know that DCT is also lined by cuboidal epithelium, but this cuboidal epithelium does not have the microvilli, brush border and the large number of mitochondria. It consists few number of mitochondria, because in DCT we can observe the reabsorption of water and some salt like sodium, but under the influence of two hormone, that is water reabsorption occurs under the influence of ADH, while the sodium ion reabsorption occurs with the help of hormone, that is aldosterone. So ADH and aldosterone responsible for reabsorption of water and sodium. It depends upon the need. Now the content here, it again becomes the hypertonic. So in last part, where the selective reabsorption occurs in the collecting duct, that is this NaCl diffuses from the common collecting duct into the peripheral tissue, and the content becomes hypertonic. So here it is hypertonic urine will be formed, because it is assisted by third process. It is termed as tubular secretion. This tubular secretion, also termed as augmentation process. This tubular secretion mainly occurs in DCT, but it also occurs in PCT and collecting tubule. But main site of tubular secretion is DCT. In DCT means some content which is escaped during ultrafiltration, and enters into the blood present in the peritubular capillary network. Now the cells of DCT and collecting duct, they use these substances, means capture these substances and secreted again into the lumen of the distal convoluted tubule, means substances like the some Na plus ions, some potassium ions, some Cl minus ions, some penicillin like content. These substances again secreted into the filtrate, and as a result we can observe the hypertonic urine. So generally the urine will be produced, it is hypertonic. So selective reabsorption is supplementary to ultrafiltration, while it is opposite to selective reabsorption. So in this way the urine will be formed in the nephron with the help of three steps: ultrafiltration, selective reabsorption and tubular secretion.